In this video, we will continue our discussion about cancer. So the topic for the today's video is tumor heterogeneity and resistance to target therapy. So this is a small topic. That's why I have two things has been included in the topic. One is tumor heterogeneity. So tumor cancer is not a result of single mutation. It is a result of multiple mutations. So before these new techniques such as NGS and different omics technology available, what people have think about the tumor is that the tumor cancer is the result of different mutation that are occurs at different place, and then they come together or agglomerate together to make its impact more vulnerable to the persons or to the individuals who are affected. So in order to explain their theory, what they have done is that they have done a study and identified different section of the tumor separately, and they have find out that HERS2 amplification is different in different section. Is different in different section. So after the NGS technology, that is next generation sequencing, has been come into existence, then they have confirmed this kind of approach through NGS sequencing, and what they have find out that the history of tumor can be found out, or the history of tumor is encoded within the cell. History of tumor are encoded within. So from the NGS sequencing, one can identify or one can detect what is the primary location at which specific gene mutation has occurred, and specific gene mutation has occurred at the what are the primary locations at which specific gene mutation occurs, and how many of different gene mutations that are combined together to form this tumor, that can be identified from NGS, that is next generation sequencing. Okay. So after so this is about the tumor heterogeneity, and next thing is the resistance to target therapy. So in this, whenever a targeted targeted therapy has been come into picture, there is always a parallel discussion about drug resistance. Drug resistance, as that what are the drugs that are being used to cure the cancer is considered as a foreign substance for the cells, and they will try to degrade that foreign substance as soon as possible. So, and in this case of cancer treatment, what people have identified is that this drug resistance has led to different gene mutations. Its mechanism leads to secondary mutation or different mutation. And that cause different issues or different problems, sometimes different kind of cancer. So this has been identified from relapsed study. Relapse study means that whenever a person get cure from the or get treatment from the cancer through a certain kind of medication, and after some time it again it got affected or some other problems occurs in the patient. So then the rebiopsy or relapse study rebiopsy has been will be done on the patient to analyze whether he is suffering from the same problem or something else has occurred within the patient. So this study has been able to find out that most or some of the in case of drug resistance, some of the drugs may lead to secondary mutation that might be more dangerous or less dangerous depending upon several factors, such as the persons on which the drug has been tested and the side effect of the drug also is one of the factors. So in this relapse study has now been considered as one of the main source of gathering information, main source of gathering knowledge so that they can have a new literature that include the drug and what are the possible mutation that might occur because of this drug resistance. So this is all about this video. Thank you for your time.